the maize trial field in mid-April. Ben Bubner and Regina Becker from the Center for Agricultural Landscape Research in Müncheberg have come to take samples from maize straw. Over a period of 3 years, a variety of maize types were cultivated here on 40 lots, genetically modified Bt maize in a comparison with different types of conventional maize. On each lot in a small strip, only the maize kernels were harvested and the rest of the plant was left on the field. Even a half year after the harvest, husk leaves, ears, inflorescences, and indeed the pistil threads of the female flowers all can still clearly be discerned. In the lab, the scientists aim to find out whether the straw of the various maize types is broken down in different ways.
Wir messen hier die Bodenatmung. Das heißt,
we're measuring the ground respiration. This means that in the soil you find many organisms, millions of microorganisms, that respire in a similar manner to ours. They require oxygen and release carbon dioxide, CO2. We can measure the CO2 here, we record it, and can make conclusions about their activity and the performance of their metabolism. These here are our soil samples. Maize meal was mixed into these soil samples, and they were then placed in these containers. Normal air from the environment flows through the samples and goes into the analyzer and is checked there. That means that the CO2 that's released by the bacteria that are breaking down the substrate is measured in that machine. In order to investigate which organisms take part in the breakdown of the straw, DNA and RNA are extracted from the probes. To do so, the maize straw is shock frozen with liquid nitrogen and can be ground very finely in a mortar. Subsequently, the maize meal is divided into portions. Purification procedures are carried out in several steps to yield DNA and RNA. The samples are analyzed with this instrument. With what is known as a capillary electrophoresis system, DNA fragments are separated according to size. This way, one may examine particular gene sequences that provide help in distinguishing microorganisms from each other. The results already are available from the first trial year. We're examining the populations of bacteria that take part in the breakdown of maize straw. We use a particular fingerprint analysis technique. At the core of this procedure is that one obtains a so-called fingerprint that consists of a variety of peaks. Each of these peaks represents a particular group of bacteria, so that one ultimately is able to declare which bacteria groups are present in the population and in what proportion they're found within the population. By comparing these fingerprint patterns, we've determined that there's no difference in bacterial colonization of transgene maize hybrids and the conventional types that we use for comparison.